All right, good morning. Welcome back. Spring break, much needed. I think summer's still like a couple more days, right? So we left off last time with Koch's postulates and went through this. I know we had talked of it before. I'm not going to go over it all again, but definitely this idea of testing specific things to see if they truly are the culprit for the respective disease has been utilized for all sorts of infectious disease. There are other opportunities to look at things like maybe compounds that could be carcinogenic, not the same idea like this, but utilizing some bacteria to see what ultimately is occurring with that. So truly, I think pretty interesting to think about these mechanisms to identify things. So there are a plethora of different organisms that cause all sorts of disease. And with that, they're all different as far as maybe how many organisms are needed to initiate the disease and how many organisms could potentially result in maybe a fatal infection. So that's what we're looking at with this. The LD50, LD is lethal dose. And in this circumstance, the LD50 is what is the concentration that ultimately will kill 50%? That's the LD50, all right? So truly depends on who we're talking about as far as the pathogens, because this can definitely be varied, all right? So as we look at this curve, and here we're looking at just whatever, like I said, pathogenic agents. It could be some sort of microbe, a bacteria, or a fungus, or whatever, or some virus. Doesn't matter, all right? And then we look on the y-axis, the mortality rate in this group. And like I said, we're looking at 50%. So how much dosage is needed to see that happen? Yes, sir.
Be like 80, yeah, 80%.
Exactly. LD80, you'd be looking at 80%, wherever you're at, whatever you're truly looking at. But oftentimes it's the 50% that we think about for evaluating this, but you could extrapolate more and get out here to maybe about the 80, and it looks like 10 to the fifth. Pretty close, give or take a little bit. So 10 to the fifth particles, whatever we're talking about, could be a dosage sufficient to get the majority. If we bump it up to the eighth, maybe, we're looking at very near 100%, right? So truly depends, like I said, on the specific pathogens for those dosages. It's not all, a boatload of them will get you. Yes.
Is there a reason we go with 50% as the norm, or is that just kind of just one example?
That is a super good question. I don't have the answer for that. I would bet there is something. I will do a little digging and see if I can get an answer for that. So when we think about this, and as we shift over to the actual infectious disease stuff shortly. We will think some more details about this. All right. But the ways that organisms can get into us, and conversely, shortly down the road here, we will see also the ways that organisms can get out of us. All right. So portals of entry and portals of exit. Oftentimes we will see these images and it's pretty similar. The ways things can get in are also the way that things could be getting out. That doesn't necessarily mean it's exactly the same, but likely. All right. And you think about something like a common cold, coughing and sneezing, spewing out some of these viral particles, coming in contact with them and bringing them into your mouth or your eyes or your nose, mucous membranes, and you've inoculated. And in turn, after you start feeling those symptoms, same thing happens, coughing and sneezing. So they got into you and they get out of you, right? So there are numerous ways that we think about. So eyes, nose, mouth. When we're thinking about the unborn child, there are potentials for organisms, potentials to cross the placenta and cause infection. Not everything, but some can. So vagina, urethra, the anus, broken skin. The skin is an awesome barrier to the outside world and provides really our first line of defense against all sorts of things that may like to take hold. And if that surface gets broken, it's open access to all sorts of potentials, and truly depends on all sorts of circumstances. Different infections can potentially result in maybe like ulcers or a lesion, maybe on your foot, that lends to something else getting in. The guinea worm, I like that one. It emerges typically out around your foot and wants to release the juveniles into the water so the life cycle can continue. But that ulcer is a wound that is open for a very long time, and walking around lends to all sorts of opportunities for organisms to get in. And in parts of Africa, infection with the guinea worm is highly likely that we can see infection with tetanus as a result of that, with the spores inoculating the wound. So openings allowing for other things are also potentials. Bites and needle sticks, all of these potentials for access in. And like I said, shortly we will see pretty much the same image for portals of exit. All right. Not always the same way it comes in, may not be the same way it goes out. So we talked about biofilms really kind of repeatedly throughout, and there are numerous microbes that have capabilities of forming biofilms. There are probably likely instances in the lab where you've seen some bacteria that look kind of slimy, kind of shiny looking on those petri dishes. And oftentimes that's an indication that they've got a capsule layer, that they're producing some sort of substance, this glycocalyx that they're secreting to the outside. And that lends opportunities for biofilm formation with that sugary cement-like material is really kind of what I think about, allowing them to stick down and truly get established in those, wherever they're at in those different parts, potentially through the implant of medical devices or catheters, lending to situations where you can get colonization, biofilm formation, and lending to issues. It truly depends on some of those circumstances. So microbes hold all sorts of potentials to get in and take hold. Capsule layers lend to some of those opportunities. Other substances that they may be producing or secreting may lend to some of these opportunities. Now, oftentimes, we classify these things that they harbor as a virulence factor, something that enhances their potential to truly establish disease, to make them more pathogenic. All right. So if they can come up with some sort of strategy to live, it gives them a leg up. So we've talked about Helicobacter a little bit before. Helicobacter pylori is a bacterium that really is kind of an oddball because it would like to live in the stomach. Most things do not tolerate the harsh environment within the stomach. Many things potentially can tolerate it long enough to move through the stomach into the intestines and cause issues. But to flat out say, this is perfect, I want to live here, is a little bit odd. All right. So Helicobacter has a handful of things that allows it that leg up. All right. So the mucin that we see right here, this mucus coating, covers the cells of the stomach and helps to protect them, to prevent the cells from being bathed in stomach acid. It's a barrier. All right. So Helicobacter comes in contact with this, and it helps it to have kind of a shield as well as it penetrates into the mucin, and then starts releasing some urease. I believe you did the urea broths to test for urease. You weren't looking at Helicobacter, but it would have turned those tubes bright pink as well. All right. So as it's producing the urease, it basically is like a buffering mechanism that helps to neutralize the stomach acid and create an environment around the bacterium that is a little bit more hospitable, a little bit more neutral, all right, giving it the potential to penetrate in, all right. As this is all occurring, probably some other enzymes like maybe mucinase that allow it to liquefy that thick layer of mucin. And then we've got an open area where the stomach acid potentially is now making contact with these cells. That's when you start feeling the issues, the burning, the ulcer gets established as Helicobacter breaks through this barrier and opens up areas where the stomach acid now makes contact. There are possibilities where it can cause bigger tissue damage, and we think of some opportunities for bleeding ulcers, but not all ulcers get as deep as that to truly cause that. Yes.
Now that H. pylori has opened that up, does that make it easier for other bacteria to enter as well, or?
That's a good question. I think for the most part, Helicobacter is the one that's got these capabilities of buffering itself and surviving. Most other bacteria aren't going to be able to do that, but a part of me is like, it's creating a nice environment right here. So could somebody else come in and be like, sweet, it's all neutralized right here, I'll take hold too? I don't know the answer to that. I think the bulk of what we truly are seeing with this is really just Helicobacter, but you never know, right? I feel like it truly could open up some possibilities for somebody else to be like, oh, well, I'll live here too, right? And that would kind of be like a sort of symbiotic situation. If Helicobacter is setting the stage for somebody else, it's a win-win for both of them, right? Not for you. So, as promised, the next one. So a lot of the same stuff, all right, as far as areas to be exits, all right. We also think about potentially mammary glands, milk, sets the stage for potentials for things to be transmitted. We can also think about immunity and all sorts of stuff, but truly that's for a little bit later. All right. Definitely some protection is conferred through breastfeeding to that little kid. But there are also opportunities for other things to be transmitted. So same idea. All right. A few extra added ones, like other stuff like potentially earwax or milk, but everything else pretty much the same as the portals of entry. All right. So we also think about, signs and symptoms as we're moving through here, and swelling or edema is oftentimes a good indication of some sort of problem happening where we're seeing an immune response. And oftentimes in an immune response, we see some sort of inflammatory process that's occurring. It's kind of a double-edged sword. Some inflammation is helpful and warranted to really kind of call in the troops, if you will. But excessive amounts of it can truly cause issues. So it depends on the level of this, all right? And anti-inflammatory agents are oftentimes utilized to try to mitigate this issue. So the microbes, lots of bacteria and other players, not to mention our own immune response and a handful of different types of white blood cells that also are releasing pro-inflammatory molecules. These truly start to establish a situation where we're trying to combat it, whatever that infectious agent is. And we can see the differences here with the one hand having some edema within there. Another virulence factor, the enzyme hyaluronidase, some bacteria harbor this capability, and it basically allows, so we've got a normal situation in our first frame, and then the bacteria harboring this enzyme allows them to break down the glue, the extracellular matrix that helps to hold the individual cells together, allowing the bacteria opportunities to move deeper into the tissues to make these infections more deep-seated. So another virulence factor, all right, allowing these cells to get in and gain entry. Collagenase is another enzyme with a similar sort of idea. In this instance, it's allowing it to break down collagen, connective tissue, making its way potentially into the vascular system, as we see here, the blood vessels and organisms making their way in. So we will see some images of gangrenous tissues a little bit later. Clostridium perfringens is a culprit for gas gangrene. And with that, this collagenase lends to the ability for that organism to get in and penetrate in deeper to start causing tissue destruction and the potential for, loss of limbs in those circumstances is truly a big concern when we think about those situations. Like I said, we'll see some images of that a little later down the road. So what we're looking at here is part of our gram-negative bacteria. And we're really looking at, remember, our gram-negatives have a plasma membrane, a very thin cell wall, and then an outer membrane that covers that little bit of peptidoglycan. That's what we're looking at here is this outer membrane, all right? Some lipid, and then some substances that ultimately, well, both the O antigen and the lipid A together, both of these are substances that the gram negatives harbor just on the outside of themselves that are big concerns with infection. Our body recognizes these substances, and in turn, it establishes all sorts of issues. It also sets the stage for questions, depends on the type of infection, potentially where we're talking about, for maybe not utilizing antibiotics to combat these. Releasing more of the antigens or the lipid over here can cause greater problems, all right? Depends on some of the circumstances, all right? So as we see this, we have substances that ultimately establish more of these pro-inflammatory molecules and fever, which also, like inflammation, is a body's mechanism to try to combat some of this. And dependent upon the severity of that, can be damaging to the host as well. All right, truly depends on those circumstances. Oftentimes, infections with stuff like E. coli, the bad E. coli, the 0157H7 that causes nasty diarrhea, bloody diarrhea. Individuals infected with this, treatment with antibiotics oftentimes makes the situation considerably worse. And so usually antibiotics are not given in those circumstances. Fluid replacement is more what they truly try to deal with, hoping that the immune response is capable of dealing with it. If you are an adult with a healthy immune system, you're probably good to go, all right? But if you're still going to have ill effects, but it's not something that you're truly, although at the time you might think I'm dying here, probably not. But if you are really little, or if you are elderly, infections with 0157 are potentially fatal as a result of the body not being able to truly combat that. And trying to treat with antibiotics just speeds up that process. Kind of crazy. So we could see here potentials for toxins to make their way into cells. And here we've got a receptor. We've got our subunits, A and B subunits for these toxins getting pulled into a cell. This is, I believe this is cholera that we're looking at with these toxins coming in. So through endocytosis, receptors snagging this molecule and then bringing it in. And once it's inside, we've got the components that ultimately cause issues at a cellular level, right? We'll see some more of that stuff. So maybe it's diphtheria, because now here I'm seeing this and it's the same A and B. So as this toxin gets in, it ultimately inhibits the cellular machinery, making it so that those cells aren't able to basically do what they should be doing. It inhibits the protein synthesis, shuts down all of this machinery, and in turn, in the process of turning off all of that, the cell can no longer function, and it's ultimately killed in that process as it inhibits all of these functions, and then spreads to another cell, and it keeps moving through there. All right. The next couple of different toxins, 2 Clostridium organisms, and they are kind of like complete opposites of what they ultimately do in the host. All right. So the top frame is botulism, Clostridium botulinum, and down below is the tetanus toxin, Clostridium tetani. All right. So different things happening with the nerves that establish different symptoms. So botulism causes flaccid paralysis, whereas tetanus causes spastic paralysis. So the first one, the botulism, muscles don't contract, all right? So it stops those contractions. And in turn, people potentially use this low levels for, removing frown lines or whatever it may be. There's other stuff that they can use it for as well. Sweating, chronic sweating, they can treat with some of that, and incontinence issues, problems with your bladder. A little of this also may help. So the opposite with the tetanus, muscles contract and they can't relax. So relaxed and unrelaxed, right? And that's the spastic paralysis. I don't think this one has the images of it. I think that's coming down the road. Different, all right? So you could see in both the first frames of both normal situations, all right? So we've got our muscle cell, we've got our nerve, we're releasing acetylcholine, making contact with the receptors on the muscle cell, and in a normal circumstance, this would cause contraction. The botulinum toxin makes it so that the acetylcholine isn't being released, and with that, the muscles don't contract, all right? Stopping it. And the opposite down below, like I said, although it's different actions, but it's an opposite as far as what's truly happening. So other molecules, glycine and GABA, making contact with the axon to stimulate the cell to relax, basically shutting off the acetylcholine. In this instance, it doesn't shut it off, all right, blocking those molecules. And it's a constant dumping of acetylcholine, keeping those muscles constantly contracted. You have all probably heard of tetanus and lockjaw as one of the ill effects that you might experience with that. More than just muscles in your face, your entire body, all right? And the images of individuals dealing with tetanus are truly very disturbing, all right? There are other issues, other disturbing things, like with botulism. Honey is something that is ill-advised to give to infants. You need to be about 2 years old before your gut is developed enough to truly deal with that, because there are potentials for some botulism spores to be in the honey. And in those circumstances, through the ingestion of that, it's bringing those microbes into the gut, and they colonize and start to grow and release these toxins. And in those circumstances, we call it floppy baby syndrome. They can't really contract any of their muscles. They can't move. They can't do anything. All right. Bad news. All right. So here's an image of some capsules. I'm scanning through here to see what do we have. It's not telling me, but my gut wants to say Streptococcus pneumoniae, because they look kind of like this, little diplococci, two of them together. And there are different types of Streptococcus pneumoniae. There are technically boatloads of strains of strep pneumo. You have probably heard of the different vaccines for Streptococcus pneumoniae, and they've got different numbers behind them. I think 16 and 26 or something like that. Those numbers are signifying strains that they're able to protect you against. So numerous types, all right? But before we think details with that, the non-encapsulated forms are types of bacteria that we would think of as avirulent or not really disease causing, right? And the capsule forming ones, they are the virulent types, right? And in turn, that shell around them hides them from the immune cells and gives them the opportunity, all right, to proliferate and cause issues. The non-encapsulated are open and available for the immune response to recognize and destroy before they ever have an opportunity to do anything in the first place. All right. So this causes issues. All right. And there were with like the ideas of looking at how microbes can potentially transmit genetic material, transformation, the uptake of naked DNA, transformation was evaluated with Streptococcus pneumoniae, looking at capsulated forms and non-capsulated forms and mixing them together, dead capsule forming ones allowed to come in contact with living non-encapsulated ones, and those non-encapsulated ones picked up the genes needed to form the capsule and became virulent, and vice versa when they tried it the other way around. But this showed the potential for transformation, all right? Not what we're talking about in this image, but that's the idea with that. So in this circumstance, the idea, a cell wants to try to make contact and recognize this, but the antigens are hidden away through that capsule layer, all right? Not allowing it to actually recognize it. There are other potentials, opportunities for the bacteria to protect itself by producing protease enzymes, protein degrading molecules that can break down those antibodies, giving it another leg up against our response trying to target. I think this is really very interesting. The flu virus is something that we see issues with every year. There are numerous strains of influenza, and we try to come up with vaccines that we anticipate will be the type of virus that we see. And we hope that we got it right. Sometimes we don't, all right? And the flu vaccine isn't as effective as we truly would hope, all right? So there are lots of things that can happen inside of your body that sets the stage for some of this variation that we're looking at right here. Antigenic drift and shift create opportunities for variation with the flu virus, giving us variations in the strains and potentially creating strains that are very different. That would be more of the shift, all right? So on the surface of these virus, oftentimes we see all sorts of little spike proteins, and these spike proteins are mechanisms that our immune response can recognize and mount defenses against. That's how vaccination is truly working in these processes, recognizing specific things. Neuraminidase and hemagglutinin are the two types of spike proteins that we see, and there are huge variations with these, all right? that set the stage for all sorts of strains of these viruses. They oftentimes have numbers associated with them, H1N1 or whatever it might be. All right. Giving you a hint. All right. So the first bit with the drift, this is something that happens that gives you small, subtle changes in that virus, a little bit of a mutation. All right. And that's what we're looking at here. All right. So the proteins, the neuraminidase and the hemagglutinin, with their specific shapes. Here we've got an altered sequence within our viral genome, a mutation that had occurred within your body, and then sneezing out this mutated form, slightly different than what you were infected with. All right. And the hemagglutinin, the shape on it, different over here, right? It's a little bit different. All right. Something different than what you were infected with. That's a bit of drift. All right. A little bit of change. The next one is more change. All right. Antigenic shift. There are definitely potentials when you become infected that you could be infected with a couple of different influenza viruses. Why not have two, right? You could, right? So here, infected with both virus A and virus B, both influenzas, all right? And as you see, their surface spike proteins are different, right? And now, in the process of these virions being inside of your cells, replicating, packaging up to be released out, there are potentials that we get a bit of recombination. We package a bit of both into one, and now we've got somebody completely different, virus C, coming out. And the spike proteins, we can see some variation, the yellow one from here and the blue one from there. Now we've got a completely different combination, right? Setting the stage for something that's maybe considerably more virulent, more of a concern, right? And oftentimes in these circumstances, we can see kind of like a fluctuation, drift kind of occurring all of the time. And then every 7 years or something like that, we see a bigger outbreak. Every year you can see individuals dying from influenza. You can see people dying from all sorts of stuff, but if we're just looking at the flu, every year people die. But then there are some years where the mortality rates are considerably greater. And it's typically after a handful of years of some drift occurring in individuals and then some shift occurring after some of this drift has happened to create a combination that is very different and considerably more of a problem. All right. And that is what we think about with this. All right. There are other things, and it's probably coming right up, that we think about stuff like maybe bird flu, right? Bird flu is a huge concern, and we think about potentials for pandemics with this, but there are things that kind of keep that in check as we think about the receptors on our cells that are needed for these virions to make contact with and be brought in, right? Typically, bird flu doesn't directly infect humans. There are some circumstances where that can happen, and oftentimes it's a situation where you've got a bunch of birds, chickens, or whatever it may be, and you are handling them, you're dealing with them. There are some opportunities where that may occur. Very rare, all right? What's more common is to have a barnyard situation, not just your chickens, all right? If you've got pigs and you've got chickens, now you've got an opportunity for these to come together and then come into you, all right? pigs and humans very similar, and receptors on the surfaces of pig cells can be super similar to our own receptors. And a lot of things can be shared between pigs and humans, infectious disease and body parts as well. All right. There are opportunities for some of that. All right. Pigs and birds have opportunities for a little of that exchange as well, with their receptors being slightly similar. Ours and birds are not. All right. And that is what sets the stage for concerns for outbreaks of bird flu, are when there are situations like that. All right. And there are situations where you may have a problem with your birds and they are dying. And what do you do with these birds? Throw them into a big pile, feed them to your pigs. All sorts of things potentially could happen. And if the pig consumes the sick bird, the potential for the pig to become infected is tremendous. All right. And if it's already harboring some flu virus, that sets the stage for something like this. All right. That's how those opportunities may occur. Did I get to the whole end of that? I did. Awesome. Okay, we will stop with that. That means a test should be happening, right? It's supposed to be. All right, we will see. I will see you Wednesday. I should have talked slower. What happened?